Ladies and gentlemen, welcome to the Ancient Data Scientist. My name is Tristan, and if you've been in the anti-aging community for a bit, you've probably heard of Horvath's epigenetic clock before. It's a way to measure someone's internal biological age as opposed to their chronological age. And on the face of it, that sounds wonderful. If we can do that, we can iterate quickly on anti-aging drugs and anti-aging regimes, get our results in a matter of months and years instead of decades. However, I personally find it very confusing what actually gets measured when we get our age according to Horvath's clock. Are we measuring the one underlying cause of aging? Are we measuring a underlying cause of aging? Or maybe in the worst case, we could even be measuring something that goes along with aging. But even if we manage to take off 20 years of Horvath's clock, we wouldn't feel any better. And to find that out in detail, today we're going to take a deep dive. I'm going to explain in detail what epigenetic and epigenetic markers means, and we're going to re-implement Horvath's clock from scratch using the same publicly available data that Horvath had in 2013. Let's dive right in. So first of all, how does DNA actually work in our cells? From DNA, RNA gets produced. It's basically a copy mechanism, but some slightly different molecule. This RNA gets sent to some other part of the cell, and from there a protein is built according to the RNA instructions. And these proteins have all kinds of different functions and actually determine what the cell does in a bit of simplified terms. So of course now we've got the problem, okay, we've got the same DNA in all of our cells, but our skin cells and our brain cells do very different things, right? So there must be some additional control mechanism to determine how that works. And these control mechanisms are called epigenetics because they control which parts of your DNA are actually active in any given cell and so what proteins get produced. And one of these control mechanisms is methylation. And it's a fairly simple control mechanism in certain terms, in that the only thing that happens is that methyl groups get attached to some parts of your DNA. So methyl is a very small molecular group, just one carbon and 3 hydrogen. So it's a very small modification, but with far-reaching consequences because the protein that actually attaches to your DNA to produce the RNA, it can't attach there anymore if there's a methyl group. So the gene that gets encoded further down the line in your DNA from where the methyl group attached, it no longer gets transcribed, so no more of the protein that this gene encodes. So methyl is a blocking mechanism, but attaching methyl to your DNA is a reversible process, so it can be done and undone again in your body. And this methylation, well, it's a very small molecular change, so you'd think it's very hard to measure. And this is the really fascinating part about this. For over 20 years, there have been very reliable methods out there to do this on a large scale. So there's this company, for example, called Illumina. I think they're the de facto standard. And they have these things called methylation arrays. And what they do is they allow you to measure the methylation at a lot of points in someone's genome. So this newest one here is for over 930,000 and older ones had less. So the one we're looking at today, mostly it was the Illumina 27K. So you would measure the methylation at 27,000 places in someone's genome. So if you look at the raw data, they look something like this. So from a singular person, you get something like this out. So here you have the place in the genome, and then you've got some value between 0 and 1. And you might of course now ask, okay, why between 0 and 1? I thought there either is a methyl group or there's no methyl group. That's because we don't do single cell measurements here. So this number is an accumulation of many cells. So 0.5 would mean in half the places there was methyl and in half the places there was no methyl. And from these data, Horvath actually built his methylation clock. And now you might of course ask yourself, okay, wow, this Horvath guy, he must have used some fairly heavy methods to get such an amazing result, right? Because it was quite the landmark paper in 2013. However, if we go to his original paper and we go to page 14, we are here on the materials and methods. And here he said, I used a penalized regression model. And this is just linear regression with some regularization. And this I personally find absolutely fascinating because it seems like this should be much harder. So to make really sure we didn't misunderstand how this works, we're going to implement it from scratch. But before we do that, I actually want to start with a baseline many of us will be more familiar with. And that is a very simple 3-layer neural network. Very easy to implement, very easy to train. But most of us have never worked with linear regression before. So I want to use this as a baseline and afterwards compare it to linear regression. Let's dive right in. It's data preparation time. So if we just use MLP and linear regression, we should be done fairly quickly, right? Wrong, unfortunately. As easy as the methods are, as hard is the data preparation in this case. We don't have any conveniently downloadable PyTorch dataset with 1 million samples, but Horvath instead used a collection of over 80 datasets. And he summarized them in this wonderful table for us. And if you look in this column here, you will see that the number of samples in each dataset is triple digits, and in many cases, actually, if you look down here, it's double digits, so fairly terrible. Luckily for you, I did a lot of the data preparation we need to actually make this work. One or two things about this table that are interesting to look at before we jump back into code. The first one is the platform. There's 27K and 450K, which refer to the number of methylation sites that got measured. Here we focus on 27K because it's perfectly fine for what we do. But of course in the 450K we've got more data. The second one is under availability. At most points is this GSE and then some number. And this indicates for us that this data set is available on the gene expression omnibus, which is a collection of data sets biologists use. And when a group of biologists does some experiments, which involve a lot of work, they can upload their data there so it can be used by someone else. And that's kind of a great thing. Unfortunately, it's kind of 90s style. It's not really comfortable to use. But we'll get there. So to jump back into code, we start by downloading all the series we want. I actually choose to include all the series that use the 27K methylation array and that were over 100 samples large. And this will result in overall a bit under 2,000 samples, which is less than the 10,000 Horvath used, but you will see that our results are perfectly fine, so it shouldn't matter too much. First, we download all our series, which are interesting, so I put this here, series names. In case you're confused, a series here refers to a data set. So one collection of data points, one group of biologists uploaded there. The nomenclature is just a bit different. Data set, they don't call this data set. But it's basically one data set. There's this library here, GeoParse, which we can use to download them. If we start the script, it will start the download process. Of course, it will take a while, so I'm gonna kill this right here now. And I'm just gonna copy paste the data in here, so we have them in here. Now comes the first unfortunate part. We can't just continue using this GeoParser. So if we just copy paste this line here and we change it up a bit, we say, okay, we instead want to now use the file path, and I love Copilot. And this should be very quick, right? Because I just open a file from a disk, but it's not, because he parses through all the samples and he does this incredibly slowly. And we can never use this when we actually want to iterate on some MLP design or whatever, because it will take like 5 or 10 minutes with all the samples to start each time. So we have to do some pre-processing. Wonderful. At the end of the day, we want a PyTorch dataset that loads from disk quickly. And to do that, to get that, we are going to create pickle files. We're going to create a pickle file for each sample. So for each patient that got measured, we're going to put one pickle file to disk, and inside this pickle file will be a dictionary, and the keys will be all the methylation sites, and of course there will be an additional key for age. So if we remember, this is what the raw data looks like, and this is what our dictionary will be like. So no problem in the end. I prepared this as a small pre-process script, which needs series names and data folders. So we're going to create some config file here real quickly where we can put this in, so we are sure that we use the same variables in all our files. And this is a boring part of data science. And to be honest, GPT wrote most of that because it's insanely boring. Unfortunately, it takes a lot of manual handling until it really works well. So we very quickly we go through our series names. We open each one from file. And then we look for some column names, etc., or if the platform is correct, so if it's really the Illumina array. We handle nan values, which is kind of important, because if you measure 20,000 things in the real world on a nanoscale, some things won't work. So there will be some nan values in a lot of data sets. in a lot of samples. We check here if it's a specific data set, because this has the age in months and not years. It's kind of annoying stuff to deal with. So if we let this run, again, this will take some time. So I'm gonna shorten this by copy and paste. It will create a folder here for each data set, and in this folder is a pickle file for each sample, so each patient. Wonderful. To make sure we got this right, let's take a quick look how they look like. So we're going to import pickle. We're going to say pickle path equals something GPT tells us. So in here, and maybe let's not take some random file, let's take the first file here. We're going to open this, and we're going to print the data. Wonderful. And if we do this, you can see down here, we got as keys our methylation sites, we got our values, and we got, of course, age. And this is the data we really want to have, which we can now quickly load from disk and use for our PyTorch dataset. Now that we've got some actual data, let's get cracking. So we're actually going to do this in two parts. First, we're going to do the MLP part, and then we are going to take a look at what Horvath exactly did. So, to get started, here's a plan. We create a PyTorch dataset, is relay MLP, this usual stuff about optimizers and loss function. We run a standard training loop, and we visualize the results. And all of this is kind of simple, but because it's a lot of code to actually write out, I'm just gonna copy paste this in here, and we're gonna go through it fairly quickly. The first thing you'll notice is that here we have a variable we don't know. That's because we need to extend our config a bit, and num probes is actually the number of methylation probes that were used. So it's not exactly 27,000, but this number, and you can actually print it fairly easily just by printing the length of the data here, and you will get the exact number, plus one, of course, because we've also got age in here. So we've got our methylation data set. We are inheriting from Torch data set. It's the usual data wrangling. We go through all the series names, we go through all the files, all the pickle files, we count how many there are. We initialize NumPy arrays and one big matrix where each row corresponds to one sample. So we have n sample rows in this matrix and one column for each type of probe. And of course a vector that just contains the ages. And here in this loop we fill this vector, we fill this matrix and this vector with the appropriate values, and down here we convert them to PyTorch and save them in the as class variables. This is not really a scalable approach because we load all the data into memory at the beginning instead of in the getItem method, but it's perfectly fine for here because we don't have that many data. Next we have our MLP, and this is basically what you get when you ask GPT to write you a 3-layer MLP. Very simple, 3 linear layers. Output size is 1 is important because our age is 1. Input size must be our num probes, and then we have some hidden layer parameters, which we can fiddle around with a bit. We use leaky relu as activation function, so instead of, for example, relu, because then we can get dead neurons, and this could lead to problems, but it's not really important. We initialize the dataset with the series names in the data folder, same as last from config. We have train size and a test size, and the split ratio is 0.8. We put a manual seed here just for reproducibility. We get our train dataset and our test dataset using random split, and we save the indices of our train and test data split. And this is actually kind of important, so we can load them later on and make sure that when we compare the MLP to what Horvath did, that we have the same train test split. So we initialize train loader and test loader. Our device is our graphics card. We initialize our model. Here we take the shape 1. We could actually just use num probes here. It's the same. We use Adam as an optimizer. We use a learning rate scheduler. You might not have heard of that before. What this does is it decreases our learning rate over the epochs. So we start with our maximum learning rate and then smoothly decrease it down to 0 over the number of our epochs. I think we have 200 epochs here and a fairly small learning rate to start with. And this will stabilize our training at the end a bit. As criterion, we take the L1 loss. I like it a bit more here, better than the L2 loss, just because L2 will give high weight to outliers. But I think for age prediction it makes more sense to not do that. Cool, and then we basically start into a standard training loop. We iterate over the number of epochs, we load our data, we put it on the device, we calculate the loss, we do the typical zero grad, backwards and step, and then for printing purposes we accumulate all the losses here and in the end divide them by the number of samples. And we step with our scheduler, but you can see it's in the outer loop here, so we only step the scheduler once every epoch. So then after we've done our training over our training data, we evaluate it on our test data, so we do basically the same in test loader, but this time we save a bunch more stuff for nicer prints. So at the end of the day, what we want to have is we want to save all errors and ages for a nice plot afterwards, and we also want to calculate not only the test error but also the test median error, because this is what Horvath actually reported in his paper. So we print all this good stuff out here: train error, test median error. We save our model and then we do just one plot which shows us how we did, and then another plot afterwards to see how the actual predictions look like. So great, let's go ahead and run this. And you can see it still takes a bit of a time for the samples to load, much faster than before, but still. Okay, and it's going, and you can see it's fairly fast. You can see the errors are steadily decreasing, and we are getting into actually kind of nice territory here. So if we if we just wait, we'll see getting it better and better. So our test median error here is kind of around 2.1, and I think it will go down to maybe 1.9. And here with our graph, and that looks really good. So we have a small error at the end. So our median error is about 1.9. Now, of course, the question is, okay, what was Horvath's error? So if 1.9 is fairly good, if I can predict someone's age with an accuracy of 1.9 years, that's a fairly useful thing. And if you go into the original paper, which is this one, and I think it's down here on page 2, and if we zoom in, we can see thus the tested error of 3.6 years indicates that DNA MH differs by less than 3.6 years in 50% of subjects. So, and DNA MH is what Horvath refers to his predicted age, DNA methylation age. So we are better than Horvath. And for me, the first time I did this experiment and saw the numbers, this was very surprising, because it seems that like this should have been harder. But the more you think about it, the more sense it makes, because Horvath used linear regression, as I said before, and that's a very simple method. A 3-layer MLP has much more fitting power, and thus we can expect better results, unless, of course, all the data that is available is very simple, and by going nonlinear, you don't get any advantage. One small thing to keep in mind there is definitely that this is for a different train test split. So Horvath used, I think he did his train test split by using different data sets. So some he only used for train and some he only used for test. And to get a better comparison, we're gonna definitely go one step forward and actually use the same method that Horvath used. If you close this here, we can also see our actual age versus our predicted age. And in an ideal world, this would all lie on a perfectly straight line. It doesn't, but you can see it's actually fairly good. So let's go right ahead and compare this to what Horvath did. Because I really don't like simplifications, I'm going to tell you in detail what Horvath did. Although it's a bit confusing, so please bear with me. Here you can see Horvath's setup. We take the same methylation data set, we do the same train test split, nothing special so far. But Horvath did not use raw linear regression, he used elastic net, which is a stupid name for linear regression with regularization. Regularization means that the objective is not just to match the data as well as possible to the coefficients that get calculated for the weighted sum, but also to penalize the size of the coefficients. So it tries to keep the coefficients as small as possible. But while this is reasonable in itself, what is maybe not that reasonable is that you choose to use a calibration function. So a calibration function is just it's just a function he came up with, and let's plot this really quickly. That looks like this. So this is not discovered or anything. This is just something he thought would enable a better fit. So it goes in some nice curve form until the adult age of 20, and then it goes linearly. And he also has an inverse function. And if we plot the inverse function, it looks something like this. But the most important part is that we plot the inverse function of the function. We just get a straight line. So so what Horvath did is actually he said, okay, I'm gonna train my fitter on the age processed by this calibration function. So from 0 to 80, it projected somewhere in a range from -4 to 4. Then I'm gonna fit my model to that, and afterwards, to actually get my age, I'm gonna take the predictions of the model and apply the inverse calibration function. And this does work. So it's kind of a classical thing you used to do before data science got big, that you would try to intuit how the data should look like to get better results, to handcraft features. Usually nowadays we don't do that anymore because the algorithms can learn for themselves. So if you do this, you can see you get a decent graph. However, you get a median absolute error of 2.58, which is worse, but maybe still fine. If you just use so first of all, if you just use linear regression, so with the easiest available thing, you take out elastic net, and then you take out the calibration functions, right? You say that you don't need it. You're just gonna do it basically raw. It's also gonna be fine. I think it's even gonna be a bit better. So remember, here we got, what was it, 2.58. So if we just take linear regression, it actually looks a bit better. There seems to be less bias. And it's 2.79. So it's a bit worse, which is maybe not surprising because we can tend to overfit a bit if we don't use regularization. However, and I'm not sure if I've tested this before, to be honest, if we just use elastic net, no, I have tested this before, and I think we need to adjust the parameters here a bit. We get a very comparable results. So I think it's slightly worse, but I think the whole calibration function thing obscures a bit what is actually happening, because it makes it much more difficult than it actually needs to be. Yeah, so here you can see we get 2.74. There's no hyperparameter tuning in here. I just tried a few values, so the results in any case are not that. If the differences are that subtle, at this point I wouldn't give anything on it. It's perfectly possible that if you do a good hyperparameter sweep, one method is better than the other, apart from what we can see directly here. But qualitatively, I would put this calibration function aside because it obscures a bit what is happening. So, but the important point is that ElasticNet gives us another advantage over pure linear regression and also over the MLP. And that is that we can zoom in on the most important methylation sites. Because ElasticNet will try to reduce the number of non-zero coefficients, and it does so quite successfully. As you can see in this graph and in this number down here, the number of non-zero coefficients is only about 1,000 from the 27,000 it could be. And actually in his paper, Horvath boiled this down further to 353 methylation sites that were most strongly correlated with aging. And he did boil this down by just changing the parameters. So if I put in here a 2 instead of a 1 and I rerun the code, I will get a test median absolute error of 2.9 years, so not much worse, but I will get a reduction to only about 600 parameters. And this is a trade-off I can take as far as I want. So I could ratchet the alpha value way up, so make regularization way more important. Thus I get less coefficients that are relevant, but I get a slightly worse result. So my takeaway from this is that while we can zoom in on the most important methylation sites, the number 353 comes a bit out of thin air. It doesn't really matter how much we take, it's just a trade-off. Okay, I've just got one final thing for you before we wrap this up, and that is the interpretation of these coefficients. Remember that all we do here is build a weighted sum to predict our age. So if a coefficient is positive at some methylation site, it means that with increasing values at that methylation site, the predicted age will also increase, and vice versa. So all in all, that means that at some methylation sites in our body, we have too much methylation as we age, and in others we don't have enough. And that's a very useful interpretation to have if you actually want to tackle these methylation errors that occur with age. So what do we make out of Horvath's epigenetic clock after we've re-implemented it from scratch? Does it measure the one underlying cause of aging? Well, of course not, but that was never an option. But does it measure something useful, something meaningful? I would say the chances are fairly high. That one of the root causes of aging is an accumulation of errors in your epigenetic expressions, specifically in your methylation patterns, seems perfectly plausible. So, and then of course this brings the next question: what shall we now do with this information? Is it reasonable to go out there and get your methylation age checked and then try to optimize for that? And I would say, of course, it's up to you personally, but if you are into experimental anti-aging methods, I would say it's a fairly good thing. It seems very solid. Also, when you do that, you probably won't get Horvath's clock, but you will get a second or third generation clock. And these are called Grim age or Pheno age, for example, and they work a bit differently, but they are quite similar, and I might make another video of them. Until then, I hope you have a great time. See you in the next video. Bye.